Homologous recombination starts with an mRNA complex binding to each of the broken ends of the double-stranded DNA. The mRNA complexes then move several hundred base pairs away from the break site. The mRNA complexes will then create an endonucleolytic cut on the strand with its 5' end facing into the break site. This fragment of DNA in between the break site and the endonucleolytic cut will then dissociate from its template strand. The same strand to which the endonucleolytic cut was made in is excised in the opposite direction to the break site using exonucleolytic enzymes in a 3' direction along this strand. The DNA fragment in between the endonucleolytic cut and the break site may still associate with its template strand. Therefore, it is exonucleolytically degraded by the mRNA complex in a 5' direction towards the break site to promote its dissociation. After the removal of this fragment and other fragments created by the exonucleolytic cuts, this creates a 3' overhang facing into the break site. A long 3' overhang is then created on each of the broken ends. Rad51 proteins then associate with one of the 3' overhangs. Rad51 proteins then search for a complementary sequence to the 3' overhang. In the case of mitosis, this can be a sister chromatid, or in the case of meiosis, this can be derived from a homologous chromosome. The Rad proteins then access a complementary DNA sequence by opening up the double-stranded DNA. The 3' overhang then binds to its complementary sequence, with the dissociation of the Rad51 proteins. This forms what is called a D-loop. DNA polymerase then catalyzes the extension of the 3' overhang, using its complementary strand as a template. Extension continues using subsequent regions of the complementary strand. Interactions between the 3' overhang and its complementary strand are called holiday junctions. The D-loop forms a holiday junction as transcription initiates. The 3' overhangs are then extended. From here, the 3' overhangs reach their complementary sequences on the opposing strand, the strand that is, which was originally broken. From here, subsequent gaps may be filled in using DNA polymerase. Here, the original strand is then repaired. In conjunction with the delivery of the gene editing technologies, scientists can also deliver a sequence with ends complementary to the sticky ends produced in a double-stranded break. This sequence will be integrated into the break site and fills in the gap in between the break site. This sequence is expressed within the cell and is chosen by the scientists to allow the modification of genes in the appropriate way. Now let's discuss gene editing technologies.